Hello students, welcome to Vivid Botany. We have matched the following type of question here. The first one is inhibitor. This kind of inhibitor is called as competitive inhibitor. Let me explain you what is exactly the competitive inhibitor. Generally they are present in the enzymes. Enzymes are organic catalysts or they are biocatalysts. They speed up the rate of reaction by decreasing the activation energy. So let us suppose this is an enzyme here. You know that every enzyme will have an active site. So this is an active site at which the substrate will bind. So this is the substrate and this is the active site that I am showing here. So what happens here, there will be an inhibitor that is exactly similar to the substrate. It is inhibitor. It is called as competitive inhibitor. So what this competitive inhibitor will do, before binding of the substrate, it will bind with the enzyme. So what it will do now, it will bind with the enzyme before binding of the substrate. So what happens here? Here, instead of enzyme substrate complex, enzyme inhibitor complex is formed. Enzyme inhibitor complex is formed. So this enzyme inhibitor complex does not allow the substrate to bind with that enzyme. As a result, what happens? The substrate doesn't convert into the product because substrate is not bound with that enzyme, so it will not convert into the product. So this is called as competitive inhibition. So we have an example here for the competitive inhibitor. The example is here, malonate or malonic acid, malonate, and the name of the enzyme is here succinic dehydrogenase. Succinic dehydrogenase is name of the enzyme, and moreover the substrate is succinic acid. So what it will do, actually when succinate binds with the succinic dehydrogenase, then succinic acid will convert into fumaric acid. But since the inhibitor malonate is bound with the enzyme, succinic acid is not bound with the enzyme, the succinic acid does not convert into the enzyme. That is the case here. But one thing we can understand is that competitive inhibition is reversible. Competitive inhibition is reversible. How we can make it reversible? It is reversible by increasing substrate concentration. By increasing substrate concentration, the things will become reversible. That is, the malonate will relieve from that enzyme and enzyme becomes free by increasing succinic acid concentration, and succinic acid can attach to that enzyme. Enzyme substrate complex is formed, and then the bond making will take place. Ultimately, succinic acid will convert into the fumaric acid. So that is reversible. Do remember that. Next, which will possess the peptide bonds? We know that peptide bonds are found in proteins. So every protein is made up of amino acids because amino acids are the building blocks of proteins. So every protein is a polypeptide, that is, it is made up of the amino acids that are bound by peptide bonds. Right. Next, cell wall material in fungi, that is chitin. We know that chitin is second most abundant organic substance on this planet. What is first the most abundant organic matter on this planet? Cellulose. Cellulose is polymer of what? Glucose. Then chitin is polymer of what? Chitin is polymer of N-acetyl glucosamine. Chitin is polymer, you can call it as homopolymer because it is made up of one kind of monomer, homopolymer of N-acetyl glucosamine. It is called as amino sugar. So where do you find this chitin? Chitin is present in cell wall of fungi. It is present in fungal cell wall. That is one thing. And next, chitin is present in what is called skeleton of arthropods. Right. Then secondary metabolite. What is secondary metabolite? See, there are one question is there. Which of the following is secondary metabolite? In NEET 2021, we have seen that question. Now we are encountering another question about the secondary metabolite here. So let me explain that metabolites are two types. What are these metabolites? There are two types that are primary and secondary. So primary metabolites are those which are produced in large quantities. They are produced in large quantity, QTY. Secondary metabolites are those which are produced in less quantity. And primary metabolites have with identifiable function. They are with a known function. Simply speaking, they are with known function. Secondary metabolites are those with unknown function, that they don't have identifiable functions. That is one thing. And what are the examples for the primary metabolites? For primary metabolites are the building components of the secondary metabolites. I mean, what do you call these? Molecules, macromolecules. So for primary metabolites, we can give the examples like amino acids, vitamins, nucleotides, etc. For secondary metabolites, we can give example like what we can give that toxins, alkaloids, polymers, terpenoids, etc. We can give example for the secondary metabolites. Now let us see the question here. Inhibitor of catalytic activity, this is malonate. I told you that it is a competitive inhibitor of succinic dehydrogenase for succinic acid, the enzyme. So malonate exactly resembles with the succinic acid and binds with the active site of SDH, succinic dehydrogenase, before binding of the succinic acid. Possesses peptide bond, that is collagen, because collagen is the most abundant protein in animal world that forms the ground tissue in the animals. We know that. At most abundant protein in biosphere is the rubisco, which is absent in plants but present in plants, absent in animals. Cell wall material is chitin, that I told you. Secondary metabolite is aberrin, ricin, that aberrin, ricin both are toxins that I wrote here. So toxins we can give example here, aberrin and ricin. And for alkaloids we can give example like nicotine we can give, caffeine we can give, and theine we can give. Theine is present in tea, caffeine is present in coffee, nicotine present in Nicotiana tabacum, and dathurin is present in datura metal that we know. Thorn apple, a member of Solanaceae, and nimbin is present in Azadirachta indica. Nimbidin is also present in Azadirachta indica, that is neem plant, we know that very well. And curcumin is a drug as well as alkaloid present in the curcuma longa, that is turmeric. And what is that? Ricin is an alkaloid present in Rauwolfia serpentina. It is used against the various snakes, some medical purposes. And atropine alkaloid present in Atropa belladonna, belladonna, that is member of Solanaceae. So from that atropine is obtained. And morphine is also an alkaloid that is obtained from Papaver somniferum. Codeine is also an alkaloid. All the drugs, it's a methylated morphine is called as modified morphine is called as codeine. So these two all can be called as the alkaloids. And polymers, we can give examples for polymers like rubber is a polymer, and so many examples we can give. Nucleic acids are polymer of nucleotides. Proteins are polymers of the polypeptides, that is amino acids. Cellulose is a polymer of glucose, and inulin is a polymer of fructose. Like that we can give examples for polymers. Terpenoids that include here diterpenoids and monoterpenoids. So these can be examples here. And in this question, if you see here, this is A2 is given for 2, and B peptide bonds collagen B4. So B4 is given here only. So this is the right answer for this question. So it is match the following type of question. It is taken from biomolecules. Every year we are encountering minimum 1 or 2 questions from only biomolecules. So please go through the content and make a note of that. Especially the tables are given in biomolecule chapter. They are very, very important. That is content, percentage of different kinds of elements in earth-type human being, and what are secondary metabolites, what are proteins, like that tables are given. One should be very perfect with those tables, and he can do very well if you are really good at those tables. So please subscribe my channel Vivid Botany. We are talking from Vivid Botany, and drop your responses in the channel, YouTube channel, so that we will discuss about them whenever we get time as per your demand. Right? So thank you very much.